Berberine is an isoquinoline alkaloid extracted from traditional medicinal plants like Berberis aristata and golden seal. For centuries, botanical extracts containing berberine were utilized in Ayurvedic and traditional Chinese herbal practices. Modern metabolic pharmacology has revealed that berberine exerts profound effects on cellular energy homeostasis. Unlike synthetic pharmaceuticals, berberine acts directly upon master intracellular signaling pathways to optimize metabolic flux. To understand how berberine works, we must examine adenosine monophosphate activated protein kinase, or AMPK. AMPK serves as the ultimate bioenergetic sensor within human cells, detecting shifts in intracellular ADP ratios. When cellular energy levels drop, AMPK phosphorylates target proteins to shut down energy storage and stimulate fuel burning. Berberine activates AMPK by inhibiting complex 1 of the mitochondrial electron transport chain. By mildly suppressing mitochondrial complex 1, berberine increases the intracellular AMP to ADP ratio. Rising AMP levels bind to AMPK gamma subunits, triggering a conformational change that promotes threonine 172 phosphorylation. Activated AMPK stimulates GLUT4-glucose transporter translocation to skeletal muscle cell membranes. Enhanced GLUT4 translocation increases peripheral glucose uptake independently of insulin receptor binding. In liver hepatocytes, activated AMPK suppresses sterol regulatory element binding protein 1C, or SREP1C. Downregulating SREP1C decreases de novo lipogenesis, lowering hepatic triglyceride accumulation and fat storage. Berberine also upregulates low-density lipoprotein receptor expression by stabilizing LDL receptor messenger RNA. Increasing liver LDL receptors enhances the clearance of circulating LDL cholesterol from the bloodstream. Furthermore, berberine modulates the intestinal gut microbiota composition, favoring short-chain fatty acid producers. Intestinal SCFA production stimulates glucagon-like peptide 1, or GLP-1, secretion by intestinal L cells. Elevated GLP-1 slows gastric emptying and enhances postprandial insulin secretion from pancreatic beta cells. Let us examine the exact molecular mechanism by which berberine regulates hepatic gluconeogenesis. Hepatic gluconeogenesis is the biochemical pathway by which the liver synthesizes new glucose from non-carbohydrate substrates. During insulin resistance, hepatic gluconeogenesis runs unchecked, causing elevated fasting blood glucose levels. Berberine suppresses gluconeogenesis by inhibiting key rate-limiting liver enzymes, phosphoenolpyruvate carboxykinase and glucose 6-phosphatase. By downregulating PPCK and glucose 6-phosphatase, berberine shuts down excess liver glucose production. Clinical trials comparing berberine to standard metformin demonstrate comparable efficacy in lowering HbA1c and fasting blood sugar. In double-blind human studies, 500 milligrams of berberine, taken 3 times daily, significantly reduced postprandial glucose spikes. Beyond glucose control, berberine exhibits potent vascular endothelial anti-inflammatory activity. It suppresses endothelial adhesion molecules like VCM1 and ICM1, preventing inflammatory cell adherence. Inhibiting vascular adhesion reduces early atherogenic plaque formation in coronary and carotid arteries. In adipose tissue, berberine inhibits preadipocyte differentiation into mature fat-storing adipocytes. By downregulating PPAR gamma transcription factors, berberine reduces visceral adipose tissue accumulation. Berberine also enhances mitochondrial uncoupling protein 1, or UCP1, expression in brown fat deposits. UCP1 activation stimulates non-shivering thermogenesis, burning fatty acids as heat energy. To maximize oral bioavailability, consume berberine alongside a meal containing healthy dietary lipids. Dietary fats stimulate bile secretion, forming lipid micelles that carry hydrophobic berberine across intestinal mucosa. Combining berberine with milk thistle extract or silymarin further improves intestinal absorption by inhibiting P-glycoprotein efflux. P-glycoprotein is an intestinal efflux pump that pumps berberine back into the gut lumen. Silymarin blocks this pump. Dividing total daily intake into 3 500 mg doses prevents gastrointestinal distress while maintaining steady plasma levels. Whether your goal is optimizing insulin sensitivity, supporting healthy blood lipids, or boosting cellular energy, berberine is a validated botanical. Combining daily berberine with physical exercise, fiber-rich whole foods, and restorative sleep unlocks optimal metabolic health. Let us analyze how berberine influences cellular autophagy and senescent cell clearance. Autophagy is the lysosomal degradation pathway that sweeps away damaged cellular proteins and dysfunctional mitochondria. Berberine activates autophagy by suppressing the mammalian target of rapamycin, or mTOR, signaling cascade. Inhibiting mTOR activates ULK1 kinase complexes, initiating autophagosome formation around cellular debris. Inside autophagosomes, defective mitochondria undergo selective degradation known as mitophagy. Clearing dysfunctional mitochondria prevents cytosolic leakage of reactive oxygen species and inflammatory mitochondrial DNA. This autophagic renewal preserves organ function, mitigating age-related chronic tissue inflammation. In cardiovascular tissue, berberine upregulates endothelial nitric oxide synthase, or ENOS, activity. Increased nitric oxide synthesis promotes arterial vasodilation, improving systemic arterial compliance and blood flow. In neuronal cells, berberine crosses the blood-brain barrier, protecting hippocampal neurons from neurotoxic protein aggregates. By inhibiting acetylcholinesterase, berberine supports acetylcholine levels, promoting cognitive clarity and memory retention. In renal tissue, berberine protects glomerular podocytes from hyperglycemia-induced oxidative stress and fibrosis. In skeletal muscle, berberine enhances fatty acid beta-oxidation enzymes, accelerating fat utilization during aerobic exercise. Clinical studies show that berberine supplementation reduces systemic C-reactive protein, a primary biomarker of systemic inflammation. Furthermore, berberine improves gut barrier integrity by upregulating tight junction proteins like occludin and zonula occludins 1. Strengthening the gut barrier prevents bacterial endotoxin translocation, protecting systemic organs from metabolic endotoxemia. In liver tissue, berberine downregulates profibrotic, transforming growth factor beta, preventing hepatic stellate cell activation. Preventing hepatic stellate activation preserves liver architecture and prevents progressive fatty liver degeneration. In pancreatic tissue, berberine protects beta cell mass from glucolipotoxicity-induced apoptosis. Preserving pancreatic beta cells maintains long-term endogenous insulin secretory capacity. Berberine also modulates circadian clock gene expression in peripheral liver and adipose tissues. Synchronizing peripheral circadian clocks optimizes nocturnal lipid oxidation and morning glucose tolerance. To incorporate berberine safely, start with a lower dose of 500 mg daily before titrating upward. Pairing berberine with a balanced, Mediterranean-style diet maximizes synergistic metabolic and cardiovascular benefits. Regular physical movement amplifies AMPK activation, creating a synergistic effect on glucose disposal. Consistently maintaining healthy lifestyle habits alongside berberine provides a solid foundation for lifelong bioenergetic vitality. Let us examine the chemical structure and pharmacological half-life of berberine hydrochloride. Berberine hydrochloride features a quaternary ammonium salt structure that gives it its characteristic bright yellow color. Following oral ingestion, peak plasma concentration occurs within 1 to 2 hours. Because berberine undergoes rapid hepatic metabolism and biliary excretion, divided daily dosing maintains effective therapeutic levels. Micronized berberine formulations and liposomal delivery systems further enhance systemic plasma bioavailability. Using standardized botanical extracts ensures consistent active constituent potency across every daily dose. Research continues to uncover new molecular targets for berberine, solidifying its place in modern metabolic science. In summary, berberine is a multifaceted plant alkaloid that targets the root causes of metabolic dysregulation. By activating AMPK, enhancing insulin sensitivity, and protecting vascular endothelial health, berberine empowers cellular vitality. Integrating this botanical nutrient into your daily routine supports long-term metabolic health and bioenergetic stamina. Let us examine the molecular mechanisms by which berberine protects against endothelial dysfunction. Endothelial dysfunction is the initial pathological step in diabetic vascular disease and arterial stiffening. Berberine inhibits reactive oxygen species generation in endothelial cells by downregulating NADPH oxidase 4. Downregulating NADPH oxidase 4 preserves endothelial nitric oxide bioavailability and microvascular elasticity. In intestinal epithelial tissue, berberine upregulates mucin 2 secretion by goblet cells. Increased mucin 2 secretion thickens the protective mucus layer, shielding intestinal lining from luminal toxins. Berberine also suppresses nuclear factor kappa B activation, reducing pro-inflammatory TNF-alpha and interleukin-6. Reducing systemic cytokine expression dampens chronic low-grade inflammation associated with metabolic syndrome. In skeletal muscle myocytes, berberine increases GLUT4 glucose transporter expression via P38 AMPK pathways. Activating P38 AMPK synergizes with AMPK to promote continuous glucose clearance post-meal. In renal tissue, berberine suppresses fibronectin and collagen type 4 production in mesangial cells. Suppressing mesangial matrix expansion protects kidney filtration architecture from diabetic nephropathy. Furthermore, berberine enhances liver glycogen synthesis by inhibiting glycogen synthase kinase 3 beta. Inhibiting GSK3 beta promotes glucose storage as glycogen, preventing postprandial hyperglycemia. These multifactorial cellular actions highlight berberine's unique position as a comprehensive metabolic regulator. Disclaimer: this video is for educational purposes only and does not constitute medical advice. Always consult a qualified healthcare professional regarding any medical condition or dietary changes. Thank you for watching Calorierecipes.